Okay, thank you. Uh, well, thank you for being here today. I'm going to start introducing you to guide docking. Guide docking basically is extra energy reward or a modification of the scoring function of docking algorithms in order to reward uh, the, the search algorithm when they positioned the atom ligand in a specific region. We have two flavors. One of the energy reward when the search algorithm positioned one atom in a specific region, and the other one is a penalization. The protein carbohydrate structure prediction is quite challenged, in particular for bigger glycans like tetrasaccharide or biggers. So as you can see in the protein data bank, less than 1% of the structures belongs of complexes between proteins and glycans. So in order to modeling this uh, interaction computationally, we use the docking algorithms, but the docking algorithms in general were developed for predicting drug-like molecules, and the carbohydrates are far away of drug-like molecules. They are flexible, they are polar, and the drug-like molecules are the opposite, are rigid, are hydrophobic. So in 2016, the group of Rob Woods developed Vina Card, and they included an extra term in the scoring function to penalize the glycosidic bond angles away from the minima. So that allows to improve the shape of the glycan during the search conformational search process. Then in 2020, the appears the glycotorch Vina. They create a branch of Vina card and included new atom types in order to modulate glycosaminoglycans. That there are particular glycans that has sulfur groups and nitro groups. So they included this and added an extra term to the scoring function. Guide docking in carbohydrate is not something new. We did it before in the past. We modified the scoring function using the idea of the water molecules in the carbohydrate binding site are mimicking the interactions of the carbohydrate in the complexes. That is logic because usually the carbohydrates, the glycans are full of hydroxyl groups and the waters are chemically very similar. So we introduce this energy reward of the position of the waters in the carbohydrate binding site, and we improved the prediction. But this allowed to us just improve the prediction of the post only for 3 or tetrasaccharides. Bigger ligands we failed because actually the algorithms AutoDock 4 doesn't allow to deal with very complex molecules. So in order to solve this issue, we modify AutoDock Vina over the previous branch of the AutoDock Vina, and we included the option to add energy rewards during the search of the conformational search. So we create a very diverse data set with 29 different proteins belonging to 22 different families and ligands sizes from tetra to monosaccharide. These are very huge ligands compared with the previous work that we did. And in order to create an easy way to select those waters that can be replaced by the hydroxyl or polar groups in the carbohydrate binding site, we developed a pipeline based on the filtering of the water molecules in the carbohydrate binding site by ideal interaction of the amino acid. Amino acid can establish ideally some interactions with the ligands. So when the ideal interaction of the side chain of the amino acid matches with the water molecule, we select those water molecules. Of course, we have some true positive and false positive, because not all of the water of the carbohydrate binding site are replaced for hydroxy groups. So here you can see that and then we introduced a positive control in which we took the real interaction of the glycans between proteins. We selected and we included in the docking scheme. As you can see, using the positive control, we reached an 80% of the accuracy predicting the right pose of the glycan at the carbohydrate binding site. Using the more general water-based, we reach almost 60% of accuracy. That means that we were able to predict these complexes between protein and large carbohydrates, outperforming the previous implementation, VinaCarb and GlycoTorch Vina. And this is not because we modify the search algorithm, this is because we modify the scoring function in the right way. The other algorithms have a similar performance in finding the right pose, but they are not scored on the first in the ranking. So they are scoring somewhere else. As you can see, our approach enrich the number of glycans of the good results, and we are using a very restricted value of RMSD to compare. Imagine these are very large glycans, so reaching values lower than 2 angstroms is very good. Here we have an example of human galectin-3, in which we selected the waters that are in yellow, and the difference between the results that you can reach using this general approach compared with GlycoTorch Vina, that was the better performance for non-biased dockings. So as conclusion, we developed this AutoDock Vina Site. This is not only for glycans. We choosing just glycan because they are a challenging system, but you can use it for any system that you want, because it allows to include energy rewards. Also, AutoDock Vina Site in this particular system outperforming the previous implementation of AutoDock Vina. And Viva is an approach that is super easy, super fast to estimate it, and was proven that compared with the positive control has a good performance, and the performance is better than the previous implementations. So thank you for your time.
Pablo, have time for a couple of questions? Yes.
Thank you very much for your presentation. Good, great talk. About your last slides, it's not clear to me the difference between those two representations, because to me the first is other type of view, the other is surface, for example. So I don't know. Can I
no,
compare?
just this is a representation of this is the apoform in compared with the oloform with the ligand, and this is just to highlight how the waters at the carbohydrate binding site matches with the hydroxy group of the ligand, and then you have the docking result. compare the crystallographic structure in light blue with the results obtained by the Torch Vina and the Vina Site.